Okay, so this kind of gives you an idea of what I hope to talk about today. I've brought my friend, the plastic brain model, to show you. I promised to show you the ventricles and what they might have looked at like when Leonardo da Vinci poured wax into the brain of an ox and created a wax casting. So I want to introduce you to the brain a little bit. I've posted slides for this lecture if you want them. There's basically going to just kind of take you through generalities in the nervous system and make sure everybody's on the same page with what's the central nervous system, what's the peripheral nervous system. If you've had this information before, bear with us. I've given you a handout on current brain imaging techniques, which I hope will be helpful in organizing your thoughts. There are also a couple of websites which are listed under lecture outlines and links in the Blackboard course menu, and you can go there if you feel like you don't have complete information in your neuroimaging technique chart. It's a great place to go to fill out that information, like what are the pros and cons. I'm going to try to mention today, I'm not sure how much we'll get to, but it's very useful going forward to understand what is an fMRI and what is a CAT scan and what can I expect to see from these two different types of scans. How are they done, and why would one be used over another in a particular situation? I have a couple little video segments which I've also posted under lecture outlines and links in case we don't have time for them. They're basically, the first one is looking at fMRI. It's an Alan Alda piece about using fMRI to understand where the brain processes language. There's a Nova Science Now clip that I would like to show on brain stimulation using magnetic fields. You can actually use a magnetic field to turn off a specific part of the brain or turn it on, stimulate it, or suppress it. And some really interesting results that have come from that. And then I'm quite sure I won't have time to show Nova Science Now Criminal Minds Born or Made, but it's a wonderful 15-minute little video that gives you a sense of where we might be going. There are other applications for neuroimaging research. This one is in forensic areas. So can you tell what a criminal's brain looks like as opposed to one who's, the brain of someone who's not a criminal? Interesting stuff. Okay, so let's see if I can. I'm not going to be able to walk around, which I don't like. Bad. Would you please turn on the bottom light? I think controls. Yes, thank you. So I have to call on you for that. So I can't really walk around because I'm tethered here to my computer. But this is the brain. It's about 3 pounds, and as you can see, it has a very convoluted surface. And if you look at the brains of different animals, they have different amounts of convolution on the surface structure. So, for example, the brain of a rat is completely smooth. The brain of a cat has fewer gyri and sulci than our brain does, but it has a few, which says something about cats, I suppose. The brain of a dolphin has more convolutions, more gyri and sulci than does our brain. The development of this pattern has to do with increasing surface area for nerve cell bodies. The outside of the brain is cell bodies, and the axons, which transmit information from the neurons, are in the inner part of the brain. So think about what it means if a dolphin has a very convoluted cerebral cortex. That means there's been an evolutionary push for that animal to have a great deal of gray matter in its brain. Dolphins are very, very social animals. It could be that's one of the reasons, that development of social capabilities. We believe that dolphins have a pretty advanced method of communication of their own language. We know that a great deal of our brain is devoted to language, and much of our cerebral cortex is devoted to interacting with our environment through vision, through movement, sensation, and the part of the brain where we see emotion is deeper in, and sort of cognition, higher thought, is mostly in the frontal lobes here. So I'll just take this apart and give you a sense, because we'll be kind of going there in a minute. And before my sabbatical, I could put it back together pretty easy. We'll see if I can still do the puzzle after being out of the classroom for a little while. But there you go. That's the ventricles of the brain. I'm going to pass it around from John around. And the bottom part here is the spinal canal. It is composed of cerebrospinal fluid. The spinal fluid is created inside the ventricles here, and then flows down the spinal canal all the way to the coccyx, and then back up outside the spine and outside around the outside of the brain, in between the skull and the brain, and that's a way of cushioning it against impact. So this is what Leonardo saw. In essence, this is the human version of what he saw when he made the wax casting of an ox's brain. So a few things to get familiar with here. I'm going to see if I can find my laser. Well, I'll use this. Okay. So the central nervous system is composed of the brain and the spinal cord. Everything that exits the spinal column, whether it exits through the skull or through the vertebrae, is part of the peripheral nervous system, is outside the central nervous system. So we make that major division between central and peripheral by simply exiting the bony case of the brain and spinal column. As soon as it's outside, it's part of the peripheral system. Here you see 3 layers which are intended to cushion the brain and allow it to float in cerebrospinal fluid, as I mentioned. We have the brain surface, which is closely followed by the pia mater, which stands for soft mother, is soft mother in Latin, actually. The arachnoid space, and then the arachnoid membrane. And Ben, if you'll go ahead and turn off that bottom one now, we can see better. The dura mater means tough mother, and it really is like a piece of thin leather. It's very tough. It surrounds the brain just underneath the skull, and it protects the brain from impact and sort of encases that cerebrospinal fluid. The arachnoid means spider-like mother, once again, the mother being the caretaker of the brain. That's why these membranes are called maters. And it's spider-like because it has these spider-like projections that sort of attach the membrane to the brain, and this is the part that's filled with cerebrospinal fluid. Thank you. So the general layout is that we have the CNS, which is the brain and the spinal cord. You see that here in yellow. And the peripheral nervous system, or PNS, and we will use those acronyms if you see them. That's what I'm talking about. That anything that's outside the skull, so that includes nerves that are carrying information out from the central nervous system and nerves that are carrying it back in. And different nerve cells, there's no neuron that does both. I hate using absolutes. I don't think there's any neuron that both carries information away from the central nervous system and brings it into the central nervous system in the periphery. So there you see it once again. This is kind of, if you were to look at the nerves, the layout of the nerves, you'll notice that there are sets of nerves that come off bilaterally off the spinal cord. There are actually 31 pairs of spinal nerves, all part of the peripheral nervous system. And when Magendie and Bell did their experiments on animals to discern which were sensory and which were motor nerves, these are the nerves that they would have been cutting during their vivisection experiments, and then seeing whether the animal had difficulty with movement or with sensation. Those were the spinal nerves that they were experimenting upon in the animal. So afferent nerves are sensory. They bring information to the brain. And remember, I gave you the convention that sensory information seems to come in generally on the dorsal side or the back side of the brain. And Patrick kindly pointed out to me that I had the word posterior and anterior on my Pure Mem, which was incorrect, but everybody got it right anyway. So thank you for that. It's an easy thing to do, to kind of mix those two up, but I wish not to. Efferent nerves are motor nerves that carry information away from the central nervous system and cause muscles to move in some way or another, or they may carry information that would stimulate a gland as well. Peripheral nervous system, everything outside the CNS, has some subdivisions, the somatic nervous system and the autonomic. Somatic is that under voluntary control. That's our sensation and movement system. The autonomic is not generally under voluntary control. It's the system that controls breathing rate and heart rate and vasodilation of veins in the surface of your body, pupil dilation, sweating. So things that you don't generally have a lot of mindful control over, although we can develop control over some autonomic processes if we wish to, if we focus on them as well. In the autonomic nervous system, we have the sympathetic and the parasympathetic division, usually known as the fight or flight. Sympathetic is the fight or flight system. Parasympathetic is the rest and digest system. So sympathetic generally turns up our heart rate, turns up our breathing, gives us the resources that we need to run from the bear, whatever it is. And parasympathetic is the system that's more highly activated during rest, relaxation, digestion. And they are always both turned on. It's a balance. In any activity, there'll be a balance between how active the parasympathetic and the sympathetic nervous system are. Whichever one is sending more signals is going to sort of control more strongly whether the heart rate is elevated or whether it's come back down to baseline. And one of the things that we see in people who are chronically stressed is that there begins to be an imbalance between the two systems. Like, so for example, when the sympathetic system is turned on, the fight or flight response is turned on, it does not necessarily come back down to baseline as it should. It might stay elevated for too long. The parasympathetic system has trouble kind of calming it down, and that's one of the reasons that we see stress-related disease is because of an imbalance in the two. There's also a very, very interesting and very evolved enteric nervous system, which is separate from these two and controls the internal organs, both sensation and movement control and sensation from the internal organs, and that's the enteric system. So we can kind of look at the nervous system branches. I like this thematic because it kind of helps you see it's branched into the central, which includes, brain and spinal cord, and here are the branches of the peripheral nervous system. It's a bit simplified, not including the enteric, but showing you that in the somatic and autonomic, both systems have afferents and efferents as well. The parasympathetic and the sympathetic are generally thought of as the efferent nerves that control what's happening in a host of organs in the body, whether they're setting you up to run or rest. Okay. So here you see a diagram that's giving you a sense of the somatic and autonomic nerves, that they're both paired nerves coming off the spinal column. And it gives you a nice cross-section of the brain, and you can start to see some of the structures there. Note that in yellow, in bright yellow, we have the brainstem. In gray, we have the cerebellum, and this is the cerebrum or cerebral cortex, the frontal lobe here. The temporal lobe is like it's been cut away. The parietal lobe is here, and that's the occipital lobe of the cerebral cortex. We'll be looking at those in more detail. A little bit of background on the autonomic nervous system, a little bit more. Sympathetic and parasympathetic nerves have, as I mentioned, opposite effects, but both are necessary. Each nerve, they're very simple in their setup. They're two-stage neural paths. So they exit the spinal column, they have a synapse, and then their next synapse is on the end organ, whether that be the heart, whether that be the eye, controlling pupil dilation, or whatever organ it is. It's a very simple two-stage neural path. This gives you a sense of what's controlled by the autonomic nervous system. You can see, and these are all things, if you think about it, these are all things that change when you are feeling stress, okay? So a couple of things to notice. The parasympathetics are shown in blue on the brain on the right. The sympathetics are shown in red with the brain on the left. And notice that all the sympathetic nerves exit the spinal column in the thoracic or lumbar region of the backbone, whereas in contrast, all of the sympathetics, is that what I said? Sympathetics exit thoracolumbar. Parasympathetics exit the brain from the cervical and the bottom end of the spinal cord. And they each innervate different organs. So the eye, there's pupil dilation with a stress response, so you can see. You can bring more light in and see what you need to see. Pupil constriction with the rest and digest response. There's an inhibition of salivation, so that's why people get dry mouth when they're public speaking, because the stress response is basically turning off digestive juices, because you need to be conserving your energy to run from the bear, not creating spit, right? So, with regard to the lungs, there's an opening of the bronchi, the airways, versus constriction, speeding up the heart rate, increasing the force of contraction in the heart. The glucose will be poured out from the liver during a sympathetic response to allow ready energy for the muscles. The pancreas, of course, which has to do with glucose balance, is going to work toward moving glycogen out of muscle and liver and into the bloodstream by releasing glucagon instead of insulin, according to the sympathetic nerves during stress. The stomach is going to be inhibited. We don't need to be digesting. The sexual response will be inhibited as well. One of my very first lab experiences when I was an undergraduate, I was at UC Davis in California, and I worked in a stress lab, which got me interested in stress. And one of our jobs was to look at the sexual response of sheep who were stressed. So my job was to actually put sheep in a truck and drive them around the little town of Davis, California, a cute little college town, and stress the sheep, and then come back, take blood samples, look at their testosterone levels, and look at their cortisol levels, stress hormone levels. I had to learn how to flip sheep, which is really interesting. You have to flip the sheep on its butt and hold its little feet up like this so you can get to its jugular vein, and take their blood samples, and then we looked at their reproductive response. And that was really the first time I became aware of how markedly stress inhibits reproduction. So why do people lose their libido when their jobs are hard or when they've lost their job? It's partly biological, because stress hormones inhibit the production of sex hormones directly. So that kind of got me turned on to the whole notion of stress and what it does and how it creates an imbalance in other systems in our body. Okay, so these are the two-stage neural paths. The parasympathetics leave from the cranial and sacral regions of the spinal column. They look like this. Their synapse is close to the end organ. So this is, let's say that's the heart. They've left the spinal column. There's an acetylcholine synapse in both at the second stage neuron and at the end organ in general with the parasympathetics. And in contrast, the sympathetics have a short first neuron and a long second one. So there's a ganglion that's just right next to the spinal cord. And a ganglion is an aggregation of nerve cell bodies that's outside the CNS. So it's a little bump in a nerve, which is an aggregation of bodies and synapses. And it really does look just like kind of like this. That's what a ganglion looks like, right? You can see it with your naked eye when you look at those spinal nerves. There's little bumps. And you know that those are going to be the sympathetic nerves exiting. There's acetylcholine at this synapse and norepinephrine at the second synapse in the sympathetic system. So we're going to talk a little bit more about this. I don't want you to worry too much about memorizing this slide or anything. I'm not going to test you on it. I want to introduce it because I do talk a lot about stress because it's my research area and I like to relate things to it. So I wanted you to have the background, but we'll talk about it in more detail actually at the very last, second to last lecture in the class. Yeah, Dorothy.
Maybe I know what does a mood state for people with bipolar? Is that in the synapse?
Yeah, no. So a beta blocker would be a drug that acts on this synapse, like the norepinephrine to the end organ synapse, right? which would then slow the effects of norepinephrine on the heart. But normally a mood stabilizer, there's a lot of different kinds of mood stabilizers. One of the most common would be a serotonin reuptake inhibitor, which we'll talk about actually very soon when we talk about neurotransmitters, which really works in the brain. So that's a drug that inhibits the after a cell releases a neurotransmitter, there's lots of things that can happen. That neurotransmitter can be lost into circulation. It can be taken back up into the neuron that made it. And the way a lot of SSRIs work is to block the reuptake of the neurotransmitter when it's in the synaptic cleft, so that there's a higher concentration of it in the synapse. So it just keeps stimulating the next neuron. That's an example of how one of them works. Now there's also other types like benzodiazepines work in a completely different manner. And that's one of the more interesting things when we talk about neurotransmitter systems, is to understand how drugs work in the brain. But we're completely outside the brain and the periphery. So the drugs that work at these synapses would have sort of different effects. The people that are on beta blockers would be on, they would be basically inhibiting the effects of the fight or flight response on their end organs by taking a beta blocker, right? So if you're on a beta blocker, you're slowing down the effects of sympathetic activation on your heart. Yeah.
Why athletes don't take it?
Because they need their heart to ramp up, right? Good point. Yeah. Why athletes don't take? I'm just making. I should repeat your questions. So pardon me if I repeat your questions so that we can hear them on the recording as well. You do not need to remember the cranial nerves. I've given you a little mnemonic for remembering it. Remembering them, you'll notice it's on old Olympus towering top, a fin, and German viewed a hawk. If you go to medical school, you will have to remember those. But basically what I want you to know is on the underside of the brain, I've totally taken my brain apart here, so I won't show you, but there are these yellow nerves, shown in yellow. There are 12 pairs of cranial nerves. The olfactory bulb is one of them, olfactory nerve, and the optic nerve is the second one. Those are big, important, obviously, sensory carrying nerves. And then the different cranial nerves generally do things in the face or head that have to do with movement. So those are important to know. It's important to know they exist. They are the first nerves that come out of the brain before you start looking at the spinal nerves, the 31 pairs of spinal nerves. So these are the cranials. Okay, let's look in a little bit more detail at how this protection around the brain works. There's, as I mentioned, this space which is filled with fluid. My son, when he was in, 5th grade, he did an experiment where, not influenced at all by his mother, of course, his science experiment was to throw eggs off the roof in little cheap Tupperware cases, and to surround the egg with different material, the egg drop thing. And he tried a few different materials. One of them was, Caro syrup, which is much more viscous than water, and which does an absolutely excellent job of cushioning the egg. The egg doesn't hit the side of the container if you throw it off the second story in Caro syrup. He tried other things as well to try to simulate, what else could the body have produced. But the point was that being bathed in fluid is a really excellent way to cushion this soft, sensitive organ. And the downside of a brain bathed in fluid is that it does knock around in that space, right? So if you're an athlete and you do get a head injury, oftentimes there's an impact not only on if you got maybe a bump on the front of the head, not there, but there's an impact when your brain kind of sloshes to the back of the skull, and the back of the brain gets an impact as well. So you get a coup and a contra coup, we call it, injury because it's bathed in this fluid. And that's the downside of having it work that way, but it's an excellent cushioning mechanism. The fluid is cerebrospinal fluid, as I've mentioned, and it's produced inside the ventricles. Here you see the human brain ventricular system, and I don't know This is a challenging 3D anatomical visualization, which is why we send you to the Visual Cognition Lab to learn your brain anatomy, because what they study is how people can understand things in 3D and rotate them and get a good feel for how they work. But ventricles are really hard, I think. They're really an unusual shape. There's nothing that I can think of in life that looks like brain ventricles. Really different. So this is the lateral view, and if I hold this up, you can kind of see there is the lateral ventricle that I'm pointing to. This is called the third ventricle that I'm holding right here that has the massa intermedia. It actually has a place where there's some fibers that go left to right through there. That's why there's a hole. And this is called the fourth ventricle. The cerebellum sits right here, and then the spinal column starts. So these are just different views. This would be the anterior view of the ventricles, and if you look, good thing it's plastic. Okay, so if you look from the top down, that's the view that you would see, and you get a nice sense that those lateral ventricles really, this whole thing is lateral ventricle, that whole sort of C-shaped structure, even the back end of it. They all have different names. The parts of them have names, but that whole thing is considered the lateral ventricle. This is the third ventricle, this little structure. The hypothalamus sits on either side of the third ventricle, kind of like that. And then the fourth ventricle, so to kind of getting a little familiar with those. There's also a chemical protection by the blood-brain barrier, which prevents certain chemicals from traveling through into the brain. We've got the skull, the meninges, and the fluid. This is the duramater. This is what it looks like if you were to dissect out a brain. You have to cut through this thick covering before you actually see the surface of the brain. And let's see. This is a little view of, so this is basically cerebrospinal fluid forms its own circulatory system. It's being produced here in an area called the choroid plexus, which is just like a little capillary network that has some cells kind of covering it to filter what goes through from the blood into the cerebrospinal fluid. And these capillary networks called choroid plexus are in the ventricles, in the laterals, and in the third ventricle. That fluid is produced, then it's flowing down the spinal cord, then it's flowing back up. So what we're looking at here is, here's the surface of the brain. Here's where the fluid has flowed back up from the bottom of the spine, and it comes out in this sort of subdural space and kind of pools there, and then it goes back into systemic circulation, kind of like lymph slowly goes back into systemic circulation. So it dumps into veins. And then so the system is always creating cerebrospinal fluid, it's always flowing, and it changes like any biological fluid. The components that are in it will change depending upon whether you're sick or healthy or what. I think I mentioned schizophrenia and inflammation in CSF. Here's another schematic. This little red stuff is choroid plexus. That's how it's spelled. It's here in the laterals. This is the third ventricle. Hypothalamus would be here, pituitary gland, brainstem, cerebellum, cerebrum. This is called the corpus callosum, just to kind of give you some landmarks to start orienting to this cross section. So the fluid, it's got little arrows. It's going down here, and then it's going all the way to the cauda equina, where the spinal column kind of comes out into a whole bunch of nerves that go into your legs. And then it flows back up on the outside of the spinal cord, so it's actually bathing that spinal cord, which is also a sensitive structure, in fluid, just like it's bathing the brain. Flows all the way up here, and then it goes into these subarachnoid or subdural spaces, and then back into circulation from these exit areas. Okay, so see if I can kind of put at least some of my brain back together so I can show you some stuff. Okay, so it's missing its brain stem, but good enough. So one of the things to call attention to here is the major fissures in the brain. So this is called the lateral fissure that you see in the diagram there, and it separates the parietal and frontal lobe from the temporal lobe. which I'm pointing to with my ring finger there. There's the cerebellum. The occipital lobe is back here. There's also a central fissure, which in this brain is right here. Kind of stand up so I can show you. That's it there. And that's what's separating the frontal from the parietal lobe. In fact, I think I mentioned that the primary sensory cortex is right here, just behind that central fissure, and the primary motor cortex is right here, just in front of it. So a few landmarks to kind of orient you to the brain. And here you see an electron micrograph of the choroid plexus. These are the capillaries coming in. It says blood vessel BV, and then this is the tissue that sort of filters the substances coming out of blood, and that's what it sort of controls what's released into CSF. So CSF is providing some nutrients. It's carrying away some waste from the brain. It does not have blood cells in it except for some white blood cells, which can be found sometimes in CSF. And it has plenty of chemical messengers in their cytokines and such. But blood cells don't go through. I wanted to review once again, just so that if you are getting confused about what's medial and lateral, is sort of easy. Dorsal, if you think of an animal, the back is dorsal, the top of the head is dorsal, anterior, posterior, and then we have the human with the same color. So top of the head, when we talk about the dorsal surface of the brain, we're talking about this. The anterior part of the brain here, posterior part of the brain, ventral is the underside. So if you were an animal on all fours, it would be like underside and the top of your head if you're in that configuration. That's what helps me remember what's what. Sections, there's different ways. We're going to be looking at a lot of brain sections. When I say sections, I mean cuts through the brain. And we'll be orienting to structures by looking at the brain as if we'd sliced it in various ways. So these are usually when you see a picture of the brain, you'll see it labeled coronal section, axial section, sagittal section. So these terms are important to get comfortable with. The coronal plane, as you can see here, I think it means crown. So it's if you were to slice the brain this way, right? And the sagittal plane is right. It's longitudinally sort of front to back. Now you can have a mid-sagittal plane, which is the view I showed you earlier, which would be cutting it right here, or you can have a sagittal plane that's left or a little bit right of the midline, all the way over to the edge. And so it's important to kind of get a sense of where you are in the various planes. The horizontal plane is also called the axial plane. So if you slice the brain this way, looked at it down from the top or up from the bottom, that's the horizontal or axial plane. Here you have them again. And this gets you a sense of what the brain looks like if you slice it in each of these ways. So here, this is the view that you've already seen where I showed you the corpus callosum, cerebrum, cerebellum, etc. That's the mid-sagittal view. In the mid-sagittal view, that's the only time you'll actually see you'll be inside the third ventricle. Because remember, the third ventricle is this thing right here. So you'll actually see part of the ventricle and not but if you were to slice it a little bit to the right, you'd get into hypothalamic nuclei and not be in the ventricle because it's quite a narrow ventricle, right? Here's a nice coronal section. These are the lateral ventricles here. This is the corpus callosum. In gray, in the darker color, you see the nerve cell bodies, or gray matter. And in the lighter color, you see white matter, which is the axons of those nerve cell bodies. So when we looked at the connectome the other day on the video from Charlie Rose, where we looked at Obama and his new brain initiative, the connectome is specifically looking at the white matter connections between neurons in the brain. So you're really like looking at where each neuron connects to another neuron. Pituitary gland, these would be the temporal lobes here. And this would be part of the frontal lobe that you're looking at, dorsal, ventral. How about looking at, I think the hardest section to kind of grapple with is the axial, because there are sort of fewer landmarks for us that way. So here we're looking at, it's in the same orientation as above. So this is it's called rostral and caudal. I won't use those words very much. I will call it anterior and posterior. But here are this is part of the thalamus, and you don't see the ventricles here. So we're at a section above we're kind of cutting it right through here, I think. So we're not really seeing much. We're seeing a little bit of the third ventricle right here. And we're in a part in the brain where the occipital lobes are sort of separated, and you're starting to see a little bit of cerebellum there. That's what that darker material is. Here's a section through the spinal cord. It has a butterfly-typed appearance. The butterfly's wings are bigger on the bottom, on the ventral side, and smaller on the top. And that's because of the difference in where the nerve cell bodies are located for the spinal afferents versus the spinal efferents, and we'll look at that. So here's the butterfly shape, the dorsal and ventral side. Note that the nerve cell bodies for the afferent nerves that are bringing sensory information into the brain are located outside the spinal cord in a ganglion. Okay, that's the nerve cell body, and then this is the end of the axon coming into synapse on another axon in the spinal cord. In contrast, the nerve cell bodies for the efferents, which carry information away, so this would be the motor information leaving the spinal cord, those have their nerve cell bodies within the spinal cord, and then the neurons exit and join the spinal nerve. So each spinal nerve has both an afferent and an efferent. And so how were Bell and Magendie doing their work? How could they, since the spinal nerves carry both, where were they making their cut? Yeah, right outside. So if you were to cut this part, right, you would only be, this is called the root, the dorsal root, you would only be slicing sensory information. And if you were to cut the ventral, then you would only be getting rid of motor information to a specific part of the body, right? So that's how they did. They basically looked at, and you can see they're very clear, these roots coming off dorsally and ventrally, and then they join together to form the spinal nerves. And that sort of explains the butterfly shape, because the bottom part of the shape, and you can orient yourself when you have neuroanatomy to the fact that, okay, I must be looking at the ventral side of the spinal cord, because this is the big butterfly wings that have the nerve cell bodies in them, right, from the spinal efferents that are carrying information out. Remember, the pia mater and arachnoid mater and dura mater cover not just the brain, but they cover the entire spinal cord all the way down. And here you see the 31 paired spinal nerves and the portions of the body that they innervate. So, for example, in the Bell-Magendie experiments, if they were to cut the nerves just from the thoracic region, they would maybe lose sensation or movement to the trunk. But when they did the lower cervical nerves, then they would have lost limb movement in the animals that they were working with, right? So the animal would have lost its ability to move its front leg, for example, if they'd gone with C6, C7, or C8, because those are innervating the muscles of the arm. Okay. It's just a nice color diagram kind of showing you where the divisions of the occipital, temporal, parietal, and frontal lobe are. And these are divisions of the brain that you'll kind of come across a little bit more when we talk about development of the brain, but they're good words to be familiar with as you start kind of reading in your textbook and such. And I'll go to the next slide because it has a little bit more. The telencephalon is the cerebral cortex, and it includes the cortex, the basal ganglia, and the limbic system. The basal ganglia are structures that are important in control of movement, and the limbic system is our emotion system. So those are all part of the telencephalon or end brain that include the cerebral cortex. The diencephalon is sort of an easy one. It means between brain, and it includes just the thalamus and the hypothalamus, which is kind of a football-shaped structure. The thalamus is larger than the hypothalamus, and it looks like two half footballs that kind of sit on top of the brain stem. It's a relay center for information coming from the brain to the periphery and back from the periphery to the brain. There's a lot of sort of second stage synapses that happen there. The midbrain is simply, or mesencephalon is just the simple two structures, this little section between the diencephalon and the hindbrain includes the tectum and tegmentum. So I don't have let me see if I have I don't have a really good representation on here, but I'll show you some other slides that show the midbrain a little bit more clearly. Hindbrain or afterbrain includes the pons, which is this structure right here. This is a brain stem that I'm holding, right? The pons means bridge, and there are a lot of fibers that cross left to right and create this bump on the anterior side of the brain stem. So that, along with the cerebellum, which actually goes this way, are the afterbrain or metencephalon. And then the myelencephalon is the medulla oblongata, which is this sort of swelling right below the pons and the top part of the spinal cord. And we'll be talking about the function and why are those important to know. They're developmentally distinct, and they also serve different functions in the brain. So we'll be kind of referring back to them. This is what a brain stem might look like if you were to kind of look at it in a nice diagram. There's the pons and cerebellum. There's the thalamus. There's the midbrain, the tectum and tegmentum. Tectum is the posterior side, and the tegmentum is the anterior side. Here is the medulla oblongata, and notice they've drawn a reticular activating system in here. This is sort of a network of neurons that is not as discrete as it's been drawn in this picture, but it's helpful to see it that way. These neurons are important in the control of sleep and wakefulness. This is a part of the brain that needs to be calmed down in order for you to fall asleep, that RAS, or reticular activating system. Okay, this is a little bit of a better diagram of the midbrain. The posterior part is I think I've got the tectum, and the anterior part is the tegmentum. The tectum has got a couple of bumps on it, the superior and inferior colliculi. Those are going to be very important as we start to talk about vision. Visual information is relayed at these two bumps. The bump is usually, if there's a bump, there's a synapse. There are an aggregation of synapses in that place, or nerve cell bodies there. Okay. This I'm going to skip for now because you'll get it when you get to neuroanatomy a little bit more deeply. We don't need to quite worry about it. I wanted to show you what the hypothalamus would look like. This in purple is it's on the side of the third ventricle here. The optic chiasm is where the optic nerves cross over, and that's shown in red. There's the pineal gland, third ventricle, corpus callosum, cerebellum. Pons is this structure right here. Okay, I'm not going to worry too much. I want to also give you a sense what the sort of internal structures of the brain look like. I'm going to go to this slide. So here you see two different depictions of some really intricate 3D representations of structures that are part of the telencephalon, right? So this is part of the limbic system, or our emotional brain, basically. This is the cingulate gyrus. It's the first part. It's the first part of the brain that sits right above. So the corpus callosum would be here. Sorry, cingulate gyrus would be this part of the brain right here on the inside. And it turns out to be very important in emotion. The amygdala is named because it's almond-shaped, and it is, notice, it's part of both the limbic system and the basal ganglia. It's basically our fear center. That's what goes crazy when you feel fear, and it's linked both with the the movement system and with the emotion system of the brain. So don't need you to memorize these terms at all. Just want to introduce them for now. You're going to get to them in neuroanatomy. Just want to have a little familiarity with them. Okay, through that. I'm going to go ahead. There's a real brain. Just want to point out, here's the central sulcus. So this would be the primary sensory cortex, and this would be the primary motor cortex here. So it doesn't look as good as as perfect as it does on the model or on the pictures. It's real. The lateral fissure, temporal lobe, cerebellum, etc. And let's go to. Okay, I just want to show you a couple of things here. There's what the amygdala, where it would be. It's in the medial temporal lobe, and this is the hippocampus. This is a very important short-term memory structure. It actually gets bigger when you use your memory. This is a part of the brain that can generate new neurons, and it does so when you use it. So it's nice to know that's also in the medial temporal lobe. And let's see, there's the cingulate gyrus that I mentioned, a nice picture. The corpus callosum is the biggest commissure in the brain where fibers go from left to right. So there's the corpus callosum in a coronal section of the brain. This is just to kind of let you know you'll be looking soon at different stained slices of the brain. And this is a stain that is taken up by nerve cell bodies. See how rich in cell bodies the outside portion of the cerebellum is. And the same thing for gray and white matter. I think I've got another. Yeah. So this is a stain showing that's about how thick the nerve cell body area is in the cortex, and the rest is white matter in this particular coronal section of the brain. Here you see the curved hippocampus in coronal section, third ventricle, lateral ventricles. This is the thalamus. That's what it looks like in this picture. So you kind of are going to get used to visualizing and thinking in 3D about these structures. Just want to introduce them. Okay. So let's talk for a little bit about how we image the brain. This brain image of Homer Simpson really speaks to the correlation between structure and function. See, his actual brain is teeny tiny, going right along with his personality, right? If you want to pull out your handout at this point, you might take a few notes about the different types of brain scans. I think that what I'll do is take a sec and show you, this will be an fMRI picture. Just to kind of start off, this is a brain scan from a 7-year-old girl who actually had a hemispherectomy at the age of 3. Difficult decision for a parent to make, but if a child has such terrible seizures that maybe start on one side of the brain and go to the other, then it makes it impossible for that child to develop normally. This is a surgery that is still done today. I actually have an acquaintance who had this done with their child, and the child's doing well, has a little difficulty with some motor function, but is doing very well. It's amazing how plastic the young brain is, and that it can take on the work of the missing tissue very readily if that type of surgery is done quite young. But you can kind of see it is a very, very striking brain scan where the whole hemisphere is gone. Let me take a sec here. Got it closed. All right. So this is Alan Alda having a brain scan. We're going to see. I'm going to turn off my mic and turn on the sound.
I'm going to be checking out my grammatical skills while I watch a video.
Yeah, we see a brain reaction within 100 milliseconds, whether you're doing a task or not, actually.
The video's narration sometimes makes sense.
A baby penguin swings on that door. The baby
And sometimes it doesn't.
The truck goes up and down in the papers over hills.
The electrodes are recording where and when my brain reacts to these mistakes. When the mistake is simply a word that doesn't make sense,
Kinga turns up the penguin really loud.
an area in the back of my brain, mostly on the left, reacts within 0.2 of a second. But when the mistake is grammatical,
The concert are starting.
my brain pounces on the error within 1/10 of a second, and this time in a region toward the front and exclusively on the left. Following me into the video booth and equipped with a much more fetching hat is 6-year-old Danica.
Throws wood into that black stove.
When there's a mistake of meaning,
Kinga claps her ball happily.
her brain, just like mine, reacts in 0.2 of a second. But when the video says something grammatically incorrect,
The pancake falls onto his head.
her brain is slower to respond than mine. And the response isn't so focused over that area in the front left. In fact, Ellen Neville argues it takes perhaps 10 or 15 years for the brain to organize itself to process grammar swiftly and efficiently in just one focused, specialized region.
Looks, for example, like that's an important area for sequencing different kinds of information, and of course sequencing is an important part of language. Looks like areas just behind there are very important for tool use, and the left side as well. Tool use, yeah.
Tool use over where language is taken.
Actually, it's possible that one aspect.
So he's having an electroencephalogram here. When your brain is active, it is, as you know, using electrical currents, and those electrical currents are detectable on the surface of the scalp. So, for example, when you're asleep, you see a very particular signature of electrical current, which is very different from what it looks like when you're awake. And in this case, they're looking at which part of the brain is active in response to this language task with a grammatical error, and comparing Alan Alda's response to that of a 6-year-old who has a slower response on the grammar issue, but not on the naming issue, which is an interesting application of EEG. So EEG picks up from your scalp the electrical impulses that the brain is emitting. It's very non-invasive. It is not expensive to do. Once you've paid for the equipment, you're just wearing a little cap and some electrodes. But it does not pinpoint with any accuracy what is going on within the inside of the brain. All it can tell us is generally which part of the cerebral cortex is being activated. It can't tell us what's going on in the limbic system or the basal ganglia or any particular structure deep within the brain. So it's not good at discerning differences in deep tissue. Just to note that we don't do standard x-rays of the brain. They're really not very helpful because brain tissue is all, it doesn't produce a differential x-ray contrast. So you'll see the skull and then the whole brain will look like the same because gray matter and white matter are not different enough in terms of the density that X-rays, they have the same ability to absorb the X-ray, so it doesn't really give us much. However, you can do a contrast X-ray, for example, a cerebral angiography where you would have somebody, taken or inject them with a substance that the X-ray would differentially pick up. Then you can get a pretty good X-ray picture of the vessels in the brain because you've created a situation where the blood is at a different absorption of X-rays. So that is helpful. An X-ray CT or a CT scan is also useful, and this is actually a pretty old technology. It came out first in the 1970s. CT scan or CAT scan is where a subject is placed in a donut-shaped X-ray machine, and basically the machine moves around the person, and a computer is combining 2D images to create a 3D image of the brain. So it's possible to detect cerebral blood flow using this technique. It does deliver a pretty, X-rays have gotten more more advanced. However, it's a high dose of radiation to give a person's brain, which is kind of crummy because you're taking multiple x-rays of the brain. So it might be done in a person who maybe had metastatic cancer and you didn't think they had a long time to live, but you wanted to know where the tumor was and if it was operable or something like that. So it does detect damage. It can detect blood flow. It's non-invasive and painless, but it really delivers a high dose of radiation, so not the best. These three methods, MRI, CT, and contrast X-rays, all look at the structure of the brain. Other methods look at the function. So they look at what the brain tissue is doing. A PET scan is an example of a functional method, and these are still used quite a bit, especially in neuroscience research. It stands for positron emission tomography, or PET. We'll just mostly use the acronym for it. In this technique, the subject is injected with radioactively labeled glucose, and then the brain is scanned, and the notion is that the more active areas of the brain are going to take up more glucose, and so you'll see concentrations of parts of the brain where more of this radio-labeled glucose is taken up, and usually you'll see these as red versus blue areas of the brain, and that gives you a sense of where glucose is being used. It helps us see what's going on in deep brain structures, but again, there's a radioactivity exposure that goes along with PET scanning because you can't do it without the radio-labeled glucose, so that's the downside of it. Functional fMRI is a beautiful technology, and magneto or EMG or MEG, also known as MEG, are good technologies that don't really hurt people as much as these others. So this is a functional fMRI scan. We are currently trying to recruit a functional fMRI faculty member to our department. We have a nice package of toys that we're offering, and we have a magnet downtown that's available, so we're hoping to bring in a really great fMRI person to our department really soon. But it gives you a sense of sort of what an fMRI scan, one thing it can do. Here in scan 1, a subject is asked to remember a face, and you see that this is the nose is up here, this is the occipital area of the brain. You can see some of the visual cortex lighting up here. And this would be part of the temporal lobe, possibly area of memory. Areas related to memory and emotion are lighting up here. Then think about the face, just this little area here, and then compare another face to the remembered face, and that's causing a lot of different integrated brain activity when we're asked to do a more complex task, and that's easily visible with an fMRI scan. So basically the way this works is the subject is kind of inserted into this giant circular magnet, and they can't move their head. It used to be that you couldn't even move a finger, but now you can do a little bit more. They've gotten more advanced with their technology. And basically fMRI is based on the difference in magnetic resonance that is emitted from different molecules when they are exposed to radio waves. So heavier molecules will have a different magnetic sort of signature than do lighter molecules. And so brain tissue looks different depending upon what kind of molecules are more plentiful in different structures. And so because of that, you're able to use an fMRI to look at the changes in activity in brain tissue. So very clear, better. These are old scans. We've actually got much better fMRI resolution now than probably these are 10 years old. Clear and detailed pictures of structure. It's a very expensive technique. The patient has to lie pretty still, and it can't be done if the patient has any metallic device like a pacemaker or something like that. So there's still some limits to it, but really an excellent method. This is the EEG that you saw with Alan Alda just now, and I need to stop, don't I? Okay, I do have a pure mem for you, so let me hand that out. So I'm gonna stop the recording.
When did that magnet downtown be available for research?
Just last.
Dennis was trying to.
Yeah, Dennis was.
I worked with them for like a while.
Did you?
He tried to get me to go to Nebraska.
Did he?
Yeah, but I'm not.
Yeah, Dennis actually recruited me to the department, so I have a lot of love for Dennis. I do. So he was hoping there would be one, but it just became available. Last spring, less than a year ago.
I guess the one that in Nebraska is probably up right now.
It should be, yeah. So, yeah, it's very
They
cool.
had to build an entire building.
We have a faculty member that's part of, I think he's part of radiology, that his whole job is running the magnet and collaborating with people. So there's new opportunities now. Okay. I threw so many terms at you today that I gave you. This is a multiple choice pyramid where I've given you the terminology. You don't have to come up with it. I don't usually do that, but I just thought, like, wow, that's a lot of new terms. So, okay.